This is Miss Pham. This video is going to be about mitosis. So our notes are going to span over 2 pages, 57 and 58. On the top of, before I go to the top, I want you to make sure that your notes are sideways. This process is long, and you need enough space to put all the pictures down as well as right below it. So make sure your notebook is sideways. You want it the long ways going horizontal. Alright, so mitosis. Again, we define this in cell cycle, and we're going to define it again. It's a process in the cell cycle that divides the cell's nucleus. Focus on nucleus. Alright, so 57 is going to be a picture of an animal cell. In this animal cell, you start with interphase. Interphase is not technically mitosis, but I wanted to emphasize that it does occur before mitosis. So if you want to make a note at the bottom, before mitosis. Okay, it's the phase where the cells grow and replicates its DNA. In between period of cell division contains 3 phases. G1S replication, or G1 is growth, S is replication, G2 is prep for mitosis. Alright, now we go into mitosis. First thing is called prophase. Your chromosomes are visible. So notice in interphase, the DNA looks like a big scribble. But then in prophase, they're clearly marked X's. So what happened is the DNA, they separate, they coil up, and they make distinct shapes so that it's easy for packing. Just like I told you guys before when we talked about DNA. Chromosomes, imagine chromosomes to be like the boxes you packed when you're getting ready to move. You have to organize everything before you make the move, or you're not going to find all your stuff and things are going to go missing. So chromosomes are visible. Nucleus starts to disappear. Chromosomes join together to make chromatids. So chromatids are just 2 copies of the same chromosome connected to each other. Centrioles make spindle fiber. Centrioles is a organelle in the animal cell that helps with cell division. Its job is to make these spindle fibers. They're kind of like ushers. They push and they pull the chromosomes and tell them where to go. And that leads into metaphase. Metaphase is where your chromosomes are lined up across the equator, which is the center of your cell. Notice they're all along this kind of center region. Each chromosome is connected to a spindle fiber at its centromere. The centromere is the center. It's the point that joins the 2 chromosomes together to make a chromatid. Alright, so notice the spindle fiber connected to the chromosome is pushing and pulling it towards the center. That's metaphase, lining up. It's very distinct. If you looked under a microscope, you could see there's these regions in the center of the cell that have kind of lined up. Then we have anaphase. Anaphase is the process that the chromosomes pull apart and split and move towards the opposite end of the poles. So in metaphase, they're centered, now they're pulling in the opposite direction. Okay, telophase is kind of like reverse prophase. Prophase, chromosomes are visible, nucleus starts to disappear, and telophase, chromosomes gather at the opposite ends and they lose their shape. So prophase, you can see chromosomes. Telophase, you start to not be able to see the chromosomes. 2 nuclear envelopes form. So we're forming the nucleus again. We got rid of it in prophase, then we brought it back in telophase. And that technically ends mitosis. So, end of mitosis. Then we have cytokinesis. Cytokinesis is the pinching of the cell and the forming of the cell membrane so that you can have 2 cells in the end. Because if you finish at telophase, you don't have 2 cells. You have 1 cell with 2 nuclei. And that's not what we need. That's way too many copies of DNA. Something must have went wrong if you have 2 nuclei in a cell. So you have to do that cytokinesis process. You have to form your cell membrane, and it has to pinch through so that you can form 2 cells. And notice I have this box in the middle, right here. Alright, this box says mitosis results in 2 identical diploid daughter cells. Diploid means 2N, which is the total number of chromosomes that you should have as an individual. Whether you're a fly or a squirrel or a snake or a human. As a human, we need 46 chromosomes. That would be our diploid number. And then tomorrow we'll talk about another vocabulary term called a haploid. But today, you need to understand that a diploid number is the total number of chromosomes that an individual needs in one of their cells. Alright, so this is mitosis in an animal cell. Let me go over it real quick. Interphase. Cell grows, replicates, and gets ready for cellular division. Prophase. Chromosomes are visible, nucleus starts to disappear. Spindle fibers are connected to the chromosome. Now, metaphase, spindle fibers push and pull chromosomes to the equator. Then anaphase, chromosomes are pulled apart. Telophase, they're pulled apart even further. Nucleus forms. Cytokinesis, 2 cells result at the end. You've got 2 distinct cells. Okay, now animal cells go through a different mitosis than plant cells, and I'm about to go over plant cells. Alright, so a plant cell goes through mitosis. Okay, interphase still before mitosis. Period of growth and replication, getting ready for cellular division. Prophase, chromosomes are visible, nucleus appears or disappears. Chromosomes join together. Metaphase, chromosomes line up in the center. Anaphase, pull apart. Telophase is the same. Chromosomes still gather on one side, and nuclear envelope forms, but they don't go through cytokinesis. What happens is a plant cell, once the nucleuses or the nuclei are made, let me find a green, what they'll do is they'll just build a wall between them. So they'll just kind of start growing and forming a cellulose wall between the 2 nuclei, and then the cytoplasm will grow and increase in volume, and now they have 2 new cells. So they don't go through cytokinesis, they just form something called a cell plate. And my apologies, this was supposed to go on 56 and 57, not 57 and 58. So again, interphase, the phase before, is called in-between phase because it's between replications. G1 is growth, S is replicating DNA, G2 is prepping for mitosis. Prophase, chromosomes are visible, nucleus disappears. Metaphase, chromosomes connect by centromere with spindle fibers, line up across the middle. Anaphase, starting to move away from each other. They're getting split at this point. Telophase, they're already split. They're losing their shape. Nucleuses are appearing, and then they're cutting them off and becoming 2 cells. Make note, these cells are still identical daughter cells. They're making 2 identical daughter cells from mitosis, and that is the whole process.